This video was produced by the U.S. Food and Drug Administration. Food. So enjoyable, so delicious, such a satisfying and essential part of our lives. And yet, do most of us ever wonder where it comes from? Where it's grown? How it's handled and packaged and shipped? Nope. Most of us don't think about any of that. Not until something we've eaten makes us sick. And then, how do we figure out which particular food was the culprit? Think about a salad bar, where you might easily eat 50 different ingredients. Each different kind of food can come from a different part of the world, and each one has different pathogens associated with it. And that means that every time you enjoy a salad, you're really exposing yourself to a large number of global pathogens, any of which have the potential to make you sick. According to the Centers for Disease Control, foodborne disease outbreaks are responsible for about 48 million illnesses, 128,000 hospitalizations, and 3,000 deaths in the United States every year. As the world becomes more interconnected, maintaining a safe food supply has become an increasing challenge, and the Food and Drug Administration has pioneered an innovative response to this challenge. Collaborating with federal and state public health labs, FDA has built a pathogen identification network called GenomeTrakr. It uses whole genome sequencing to look at the DNA fingerprint left behind by disease-causing bacteria, allowing FDA to identify the source of an outbreak with more detail and clarity than ever before.
Whole genome sequencing is much like a radar gun to a police officer or a state trooper who's looking for speeders. Whole genome sequencing will now be able to surveil the food supply for pathogens, much like a radar that runs from the surface of the ocean to the edge of space. We'll be able to detect outbreaks when they happen early, and we'll be able to stop them in their tracks before they become a public health crisis.
The GenomeTrakr network has 3 different elements to it.
The first is just to build a database of known pathogens from known foods, from known geographic localities. We know from the examples that we've tested that you can tell salmonella whether it's from California or from China. And that information can help an investigation in determining where the contaminants entered into the food supply.
So at FDA, of course, we have a very large surveillance program, and we, of course, test many foods that come in at ports of entry, and we test many domestic foods before they go into the food supply. Sometimes we find pathogens like salmonella or E. coli or listeria, and when we do, we now sequence them to see if they're related to a foodborne outbreak event, and then more so to see if we can track it back to the very source from where it came.
Pathogens evolve very quickly and have thousands of genetic variations. After spending time in a particular geographic location, a pathogen like salmonella begins to acquire unique genetic signatures, like fingerprints, that identify it as coming from that location.
One good example of where whole genome sequencing has shown its really unique power was in 2012 when we had an outbreak related to spicy tuna associated with sushi.
So we knew people were getting sick, and we knew that some of them said they'd eaten at a sushi place, but we didn't know which ingredient was responsible. In fact, initially they thought it was in the hot sauce. And so what we did was we sequenced a dozen isolates from our freezer, from food from around the world that were related to this particular bacterium.
When FDA compared the bacteria from the patient back to the samples in GenomeTrakr's database, the culprit turned out to be a pathogen called Salmonella borrelia.
And unfortunately, Salmonella borrelia can be found all over the eastern hemisphere of the world, many different places. And our current forensic tools were not able to distinguish the source. When we applied whole genome sequencing to those isolates of Salmonella that we collected from all over the world, we were able to link it directly back to reference isolates that came within 6 miles of the actual tuna facility in Southeast Asia where the firm that was contaminating the food supply.
This was a major finding because it showed that pairing genomic information with geographic information could narrow the search for the source of a contaminated ingredient, even when the source was located halfway around the world.
We never knew that sequencing isolates from a port of entry that were collected 5 years before that event would tell us the potential source of the outbreak.
And whole genome sequencing is also telling us things we never thought we could learn about bacterial pathogens.
Things like certain Salmonella are now resistant to heavy metals, and they live in tuna and other top predators in the sea where heavy metals accumulate. These are adaptations or changes in the germ that we never knew existed. And by learning how the germ itself is evolving and changing, we'll be able to come up with better preventive controls for the food supply and for food manufacturers to stop the contamination in their plants, in their facilities, before it spreads.
In 2014, the GenomeTrakr network played a role in a regulatory action by the FDA and traced the source of a multi-state listeriosis outbreak right to the firm that made the contaminated product, a queso-style cheese.
Their pasteurization process was not working correctly, so the cheese wasn't being sterilized, if you will, to keep the pathogens out. And once the listeria established itself in the facility, it was in everything. And so almost every batch of cheese was contaminated with listeria. So anyone that had a particular susceptibility to this organism was getting sick.
FDA was able to sequence the isolates of listeria from cheese collected from the firm and compare them with isolates that CDC had collected from the sick patients.
Typically what happens is people get sick, they present themselves to a physician, and the physician says, oh, you have disease X, Y, or Z, in this case listeriosis. It's fairly rare, so physicians know to upload the information. In this case we would sequence it, and then you start comparing the information. What was interesting about this case is in the past this would have occurred over a series of months. Now, because of digital sharing of information, we were able to move much faster with this type of technology, and so we can now say, oh, well, instantly we have a match, much like a criminal case where you have DNA evidence and you can link a criminal to a particular scene. We were able to link a food to a group of people that were getting sick, and this was the first time this was done.
Whole genome sequencing strengthened FDA's evidence linking the outbreak strain to these cheese products. FDA was able to shut the facility down, and then a different outbreak occurred.
We noticed that there were illnesses associated with peanut butter or nut butter, and they were in different states, in different parts of the country, and it was not clear that they were related. At the same time, FDA field inspectors had done a survey of a plant that made nut butter and found Salmonella in the facility. When we sequenced those Salmonella and put them in the database, they were almost perfect matches across 4.6 million data points to the illnesses in different parts of the country. So we had actually used it in this case to detect the source of an outbreak just as the outbreak was starting. We were able to stop the food product from going further into the consumer's food supply, and we were able to stop the outbreak before it advanced to many dozens or hundreds of illnesses.
The second element of GenomeTrakr is collecting global samples.
Getting federal, state, and international partners collaborating to build the database, because bacteria are globally distributed and food is globally traded. So any food you might buy from your local grocery store or eat at a local restaurant may have come from anywhere around the world. And so we need global collections to also be included.
Surprisingly, it doesn't really take that much time to collect all this genomic data.
Takes a little less than a week. And the process has many components. The first is that actually it's isolating the bacteria from the actual food. So for example, the way we soak a tomato and get the bacteria out of it is different than how we treat avocado or peanut butter. There's a whole group of the FDA that specializes in how best to get pathogens out of different kinds of food. Once you have the pathogen, then it's relatively straightforward to produce DNA and then do the chemical reactions to build a genomic library to sequence that. And then there's a process of the data analysis. We move the data from the sequencer into a high-performance computer, and then we do data analysis and then give those results to a subject matter expert who can assess the results and provide an interpretation.
And the third element is sharing all this data, which is housed at the National Institutes of Health.
Data is publicly available. Anybody can see the data, screen the data, and can compare their particular bacterial pathogen to the database to see and characterize what pathogen they've discovered.
Not only are we making the data and metadata public in real time, we're also making the data analysis public. Our partners at NIH are building an analysis pipeline off the database. So the genome data comes in, the new genomes are matched against the existing database, and a tree is built revealing the evolutionary relationships of all the bacteria in the database. As you can imagine, the tree is huge right now. And so when a new isolate comes in, it's like a new twig being added to the big evolutionary tree of pathogens. The applications of this are probably something that we can't even foresee right now.
But the actual genes and the actual variation that is arising in these pathogens might be used for many other useful tools. For example, new vaccines, or new understanding of pathogens, new ways to control bacteria to get it out of, say, a farm or a manufacturer's facility. By making the data publicly available, we allow a larger group of people to see it and explore it and then come up with new ways to use it.
And the FDA really does want a larger group of people involved. They're very interested in finding new state, national, and international partners.
We can help in many ways. We have a large set of resources as well as the experiences of setting up a network, and so we're happy to help you however you would like us to. That may be education, that might be sequencing isolates, or that might be just integrating you in our data analysis process. We're happy to work with you in any way that we can help.
GenomeTrakr is just in its infancy, but already it has had a profound impact on food safety. It began with a database of information on pathogens like Salmonella, Listeria, and E. coli. But in the years ahead, it will include data on any bacterial or viral pathogen, not only foodborne pathogens, but infectious agents like tuberculosis, SARS, hospital-acquired infections, and even those affecting animals. By bringing whole genome sequencing into the laboratory, FDA, along with its growing numbers of domestic and global partners, is truly pushing back the frontiers of outbreak response.